Biology students, this is 9.2. The main idea of mitosis and cytokinesis is eukaryotic cells reproduce by mitosis, the process of nuclear division, and cytokinesis, the process of cytoplasm division. During mitosis, the cell's replicated genetic material separates and the cell prepares to split into two cells. The key activity of mitosis is the accurate separation of the cell's replicated DNA. Mitosis increases the number of cells in a multicellular organism as it grows, and it replaces damaged cells. Prophase is the first and longest stage of mitosis. It's the cell's chromatin that tightens and condenses into chromosomes. Its sister chromatids are structures that contain identical copies of DNA, and the sister chromatids are attached at the centromere. So if you remember, sister chromatids look like V's when they're apart, look like X's when they're together, and being held together by a centromere. As prophase continues, spindle fibers, right here, spindle fibers, centrioles, and aster fibers, and centrioles, form a spindle apparatus, and this apparatus attaches itself to each of the sister chromatids before cell division. The second stage of mitosis is metaphase, and it is the shortest phase. Sister chromatids are pulled along the spindle apparatus towards the center of the cell, which causes them to line up in the middle. I think when you first start this part, this is probably one of the easiest phases to see in a microscope. So during anaphase, as you can see, the chromatids are pulling apart or away. For anaphase, the microtubules of the spindle apparatus begin to shorten as they're pulling up. The sister chromatids separate, so they're no longer X's but V's, and the chromosomes move towards the poles of the cell. Telophase is when the chromosomes arrive at the poles and they begin to decondense. So if you look here, the DNA I have right here is the same exact DNA I have over here. They're twins. It's not any different. So the only difference between anaphase and telophase is that the chromosomes are decondensing and there are two new nuclear membranes, these purple pieces, begin to form and the nucleoli reappears. The spindle apparatus disassembles and it goes away. So we're done with mitosis, right? Well, there's this little matter of cytokinesis. After the two daughter cell nuclei have formed, cells have to undergo a completely different stage called cytokinesis. In animal cells, microfilaments constrict and pinch off to form two cells. Plant cells are very different. Instead of pinching in half, a new structure called the cell plate forms between the two daughter nuclei.
Cells reproduce by a cycle of growing and dividing, which is called the cell cycle. The cell cycle has three main stages: interphase, mitosis, and cytokinesis. Let's examine the cell cycle for a typical animal cell. Interphase is the longest stage in the cell cycle. During this stage, the cell grows, replicates its DNA, and carries out normal cellular functions. During mitosis, the cell's nucleus and nuclear material divide. This stage of the cell cycle is divided into four segments: prophase, metaphase, anaphase, and telophase. Prophase is the first and longest stage during mitosis. During this stage, the cell's replicated chromatin coils into pairs of sister chromatids. The nucleolus disappears, and spindle fibers begin to form in the cell cytoplasm. Toward the end of prophase, the nuclear membrane disintegrates. The spindle fibers extend from the centriole at either pole of the cell and attach to the centromere of the sister chromatids. During metaphase, the sister chromatids are pulled by motor proteins along the spindle apparatus toward the center of the cell, and they line up in the middle or equator of the cell. During anaphase, the chromatids pull apart. The microtubules of spindle fibers begin to shorten, pulling the centromere of each sister chromatid, causing the sister chromatids to separate into two identical chromosomes. During telophase, the chromosomes reach the opposite poles of the cell and begin to decondense or unwind. Two new nuclear membranes begin to form, and the nucleoli reappear, and the spindle apparatus is disassembled. Finally, cytokinesis is the stage where the cytoplasm divides, creating two new cells.
Make sure you're studying for test 9.2 and be able to explain what the events of each stage of mitosis are and in order, and the process of cytokinesis, both for plant and animal cells.